thing on? Are you working or are you packing? There's cells all over your body. You're made of all these tiny little cells. Everyone knows this. The study of this is called microbiology because it's microscopic, okay? And your cells are all doing different things, okay? Some of them are working and others of them are making more cells, okay? We have an analogy here with human beings, okay? We got human beings that are working, but when you have a baby, you got to take some time off from working, okay, to make that baby. Now, we got baby-making cells that are called stem cells, and there's a lot of different cells that actually can make new cells, and they are located at the base of your skin, and they are located in the center of your bones. The bone marrow is what's responsible for making new immune system cells and red blood cells when you lose blood or need to replace blood after you donate blood, or your immune system is oftentimes fighting different things, so you need to be replacing cells, and that's kept in a super safe vault in the center of your bones called bone marrow. And there are a lot of stem cells there that are going to be going through mitosis, that process of making new cells, new diploid cells that have a full set of DNA, 23 pairs, 46 total for human beings. That's one cell just dividing into two. Now there's a process there, prophase, metaphase, anaphase, telophase, where it talks about how you're going to separate those chromosomes perfectly in half. But for the purposes of this lecture, just know we got DNA replication happening before that, so you have twice as much DNA going into this process so that when you split, both cells have a full set of DNA. Okay, so your original cell has to replicate its DNA, then split the DNA so that this cell is diploid and this cell is diploid. They have a full set of DNA each inside of their nucleus, that 23 pairs, 46 total, okay? Now, this is gonna happen a lot when you're an infant and you just need to make more cells to grow bigger, okay? This happens anytime you have a growth spurt. This is happening all the time whenever you need to replace cells inside of your mouth, because skin flakes off because you're eating stuff. Your digestive system, you lose a lot of those skin cells, because your digestive system is internal skin. But there are other places in your body too. You just need to make more bone cells. Occasionally you make new liver cells, new kidney cells, all those different things. Okay. The spot where we don't have a lot of mitosis happening, where we are not making new cells regularly, is your heart, your muscles, and your brain primarily. These are cells that we are in G0. These cells are doing work. They're doing their job. These cells are making enzymes, which are a type of protein, in order to do the work. And the enzymes are powered by ATP, which is made by the mitochondria doing cell respiration, okay? But these are just cells living their life, doing their job. Your brain, every time it's thinking, it's growing and getting better. You're not making new brain cells. You're working with the brain cells that you already have, and they're growing and becoming better at their job and expanding. And that requires a lot of enzymes to do work. When you eat food, your stomach, your digestive system, pancreas, liver needs to create enzymes to break that food down and then absorb that food. That's work, okay? These are G0 cells. They can't be making new cells. We don't need to make new cells at that point. What we need to do is do the work of breaking food down, of detoxifying your blood in the case of the liver, of filtering your blood in the case of the kidneys, of digesting your food in the case of the stomach and parts of the small intestine, as well as the salivary amylase that you make inside of your mouth. So that enzyme, that salivary amylase that breaks down carbs in your mouth, that is something that a G0 mouth cell is going to be making and secreting in order to do the work that it needs to do to help your body function. Okay. Now, this process, these cells all across your body that are doing work, they're said to be specialized. They are developed. They have, this is kind of like for human beings, this is like after you go to college and you start to specialize in your particular field. You're not just a blob. You are really good at doing this, making that, right? You are the stomach cell that's really good at making digestive enzymes. You are the pancreas that's good at making digestive enzymes. You are the brain that's really good at receiving signals and sending electrical signals, releasing neurotransmitters and hormones. You're really good at your job. So you're specialized, okay? You're very specific. And that specialization and that work basically means you're just making proteins all day long to get the work done. It takes a couple minutes to make a protein. And the process is officially called protein synthesis. And your DNA, which is the instructions to make proteins, kept safe inside the nucleus. This is your permanent copy of all of the instructions to make proteins. We've got 3 different types of proteins we could make. We've been focused on the enzymes, which are the working proteins, but there are also structural proteins that make up the cytoskeleton, like actin is the most common and the most common. There's also signal proteins they're hormones. They're called peptides. They're really small proteins often, and you could put those out into the bloodstream when you need to send signals and coordinate behaviors. This is how your stomach tells your brain that you're hungry or full or upset or something like that. So there's all these signals that will go through the bloodstream, okay? But they're all proteins. And you make a protein through a pretty basic process called protein synthesis, where you transcribe the DNA into mRNA, then you translate the mRNA into a protein. Proteins are made of amino acids, and you have to string those amino acids together. That work is done by a ribosome, and the ribosome is helped by a tRNA, which is bringing the amino acids to the ribosome so the ribosome can put them together. So the ribosome is there reading the mRNA, 3 letters at a time, it's a codon, and it's reading that, and then it's taking the amino acids and putting them together in a string, and those amino acids strung together fold up into a functional working protein, whether that protein is an enzyme, a cytoskeletal actin, or a hormone. You are able to make that protein through the process of transcription and translation. The ribosome making the protein needs a tRNA to bring amino acids to help, and the ribosome is reading the mRNA. The mRNA, I like to say, is the temporary instructions to make a protein, and that was made through the process of transcription, which occurred in the nucleus. Transcription is when the DNA is opened up by RNA polymerase, which is an enzyme because it's doing work. The DNA is opened up by RNA polymerase, and then RNA polymerase will build mRNA on top of the exposed DNA, 1 letter at a time, to copy that information. So your mRNA being a temporary copy of the instructions to make DNA is built from the DNA, letters on letters, okay? The DNA, being the permanent instructions to make all proteins, is filled with genes. A gene is the permanent instruction to make one protein, okay? And the DNA is just a collection of all the genes, like a cookbook is a collection of recipes, okay? The rough ER is a structure attached to the nucleus that has a bunch of ribosomes on it. That's why it actually looks rough under a microscope. And so ribosomes could be reading the mRNA right here in the rough ER structure, and then the finished protein will go to a 3-bag structure called the Golgi apparatus, where you have packaging and shipping, where the finished proteins will be sent off to where they need to go. Maybe that's another spot in the cell, or maybe it's entirely outside the cell, like in the case of secreting saliva in your mouth. That's outside the cell. You're making an enzyme for outside of the cell. And the Golgi apparatus is the packaging and shipping of those finished proteins, okay? This is all of our working cells. If you're working with DNA, you're doing transcription, you need to have the DNA unpacked in chromatin form. And this unpacked is easier to open, easier to read. Now the packed up DNA, the only time you would ever pack up anything is to move it. Not to read it, not to use it, not to transcribe it, but to move it. So when you pack up DNA, it's called chromosomes, okay? Now you'd only pack it up ever if you're going to move it, which we mentioned at the beginning, is when you are making new cells. So when you have to make new cells through the process of mitosis, there you're going to pack up all of your DNA into chromosomes and line them up in the middle and then separate them, and then you have 2 different cells there, Okay, so those packed up, that packed up DNA is not easy to read, but it'll unpack once we put it back. Once we separate and we put it back inside of a nucleus and protect it, it can unpack, and then you can start to use it again and do your job of working as a G0 cell. Okay, now the other type of cell division to make new cells is called meiosis, and this is a very specific type where you're not making normal diploid cells from diploid cells. You're making haploid cells from diploid cells. Now, the only reason you would ever want a cell that only has half a set of DNA is if you are planning to then maybe combine it with another cell that has half a set of DNA later through the process of fertilization. And this is combining the egg and the sperm for sexual reproduction. Okay? When you fertilize the egg with a sperm, both of them are haploid. They have half a set of DNA, and together they become diploid with a full set of DNA, and that first cell is called a zygote. Okay? That cell is going to go through a whole bunch of mitosis in order to become a embryo, fetus, baby, right? But at the very beginning, the process of making gametes that occurs in your gonads is called meiosis. Okay?